Hi, everybody. So, how's everybody doing? Just want to say thank you for waiting until the very last session. That takes a lot of staying power. But what I'm going to do today is I want to talk to you about the work that we have been doing, studying the most unique of all mammalian species. It's not humans, it's bats. And Brian Kennedy touched on the fact that we're going to need to find new ways to really try and extend human lifespan and health span tenfold. And I hope that I can convince you today that studying bats is going to hold the answer to that. Now, as we know, hold on. Oh, wrong one. That's a vampire bat, by the way. So, two huge grand challenges that society now faces today, as we all know, is healthy aging. We know this graph that by the time we are 60, there's a 70% chance of having at least one, if not multiple, diseases of the old age. And we've got to find ways to extend our health span to our now extended lifespan. We know all this. But the second, it's something that I think we need to talk about a lot, is no matter, you can live long, but if you can't find ways to be able to deal with infectious diseases, then the diseases, the pathogens, they're going to get us. So we need to find new ways to deal with infectious diseases. And we've all lived through, thank God, the COVID-19 pandemic. And we've realized the havoc that a new virus introduced into the human landscape, a new zoonotic virus, can cause. And with an estimated 12 billion zoonotic events every single year, so a virus hopping from one species into the next and trying to establish itself, we need to find new solutions to deal with the future inevitable pandemics. But we're going to be all right. We're going to be all right because nature, she's already found solutions. And these solutions lie in bats. Now, what I'm going to do today is indulge a little bit in how beautiful our bats are. So in case people don't know, 1 in 5 of every living mammal species on this planet is actually a bat. There's lots and lots of them, and they're extraordinarily beautiful. So look at this fellow. Anybody want to guess this guy's name? It's called a wrinkled-faced bat. Look at this beauty. Only a mother could love him. It's a vampire bat pup. Very, very pretty. Then you have these beautiful nectar-feeding bats. This is a bat that's feeding on these banana trees. They're really, really important for pollination and seed dispersal in the tropics. Look at this beautiful bat. Anybody guess the name of this bat? It's called a northern ghost bat because it's white. It's one of 2 white bats. Then you have this epimorphus, another bat. Wings are very, very important for bats. They're the only mammals that have achieved true self-powered flight. This is a barbastella, one of my favorites. Fantastic picture. Bats do drink water. And this is a Nyctarius thebaica. And again, just look at the wing formation. But bats, they're beautiful. They're very, very important. There's one that lives in a tent, and it makes a tent out of leaves in a tree. There's another fellow up close to you, just in case you want to see what a bat looks like up close. But why are they important for aging and disease resistance? Well, as you can see, this is a chart here it was originally designed or conceived by Steve Ousted, whereby you can actually predict how long a species can live for given its body size. And typically there's a correlation. Small things live fast and die young. Big things live slowly and live long. But bats buck the trend. The smallest mammals in the entire world are bats. Bats can live way longer than expected given their body size. Indeed, the longest lived species, Meotis brandtii, caught as an adult, recaught 41 years later. That is the equivalent of about 250 to 280 human years, if you correct for body size. And they do not show any signs of aging, and this is in the wild. And the majority of bats live way longer than expected given their body size. But bats are also very resistant to multiple different viral infections. You can infect bats with SARS-like coronaviruses, with Hendra, with Marburg, which is close to Ebola, and they don't get sick, and you can infect the cells, and they also have a very different immune response. So bats have evolved mechanisms of modulated their own innate immune system and potentially adaptive immune system to be able to deal with viruses, to eradicate them, to remove them, and to not get sick. So studying bats can solve those 2 problems we have. So work that I've been doing now way longer than I want to think about it, maybe now even close to 30 years, is to try and develop bats as a new model study system to be able to uncover molecular pathways that underlie extraordinary aging and also disease tolerance in mammals, and to use that information to help translate into humans to find novel solutions for ourselves. Ultimately, you want to ask the question is, how do bats defy the aging process and also limit disease? But you're going to have to make a toolkit, because we didn't have model organisms or model systems. It wasn't like humans, it wasn't like C. elegans or mice, where already all of the technologies and the tools were pretty much had been put in place over the past 100 years. So we had to do it ourselves. So the first thing you had to do if you want to develop a new model system is you really need to uncover the underlying DNA code that leads to the unique adaptations and the wonders that those species can achieve. So, of course, we needed exquisite genomes. So myself and Sonia Verns, we co-founded Bat1K, which is a genome sequencing consortium to sequence the genome of every single living bat species on this planet. But we wanted to sequence these genomes to reference quality chromosome-level assemblies, so we could really start to interrogate and understand the unique adaptations that bats have evolved. We have a whole group of people. This is a global movement. There's over 400, 500 people as part of our Bat1K consortium. We published our first paper in Nature showing how can you do it, how can you sequence the DNA, how can you get the DNA, how do you move all these things around the world. It got published in Nature in 2020. But this was our pilot study. And what we've done is we've broken up the approximate 1,500 species of bats now into different phases of study. Pilot's done. We've moved now on to phase 1, whereby we've sequenced a representative of every single one of the 21 bat families. These are species from all around the world. Some unique species found only along the river Kwai in Thailand, who's Crassonectris. But we've sequenced a representative of them all. Indeed, what we have now are over 100 reference quality genomes from every single bat species that are sequence array to analyze. We want to bring all of the communities together. So if you guys are interested in the cool stuff bats have evolved, please join us in the analysis of these genomes. But again, then you had to ask the questions, what actually happens to bats as they age? What changes? You know, it's a bit facetious here, but someone goes, is that really a problem? And I was like, I don't know, actually, that part. But what happens to bats as they age? So we had to develop the methods to be able to study the longest-lived bats. And the longest-lived bats do not do well in captivity. We need to study bats in the wild. The longest-lived bats are typically temperate insectivorous bats. So working with Sebastian Puschmann and Frédéric Touzelin, we were able to develop this system, and I'm going to show you this little quick movie, whereby we had a population of the longest-lived myotis bats that were found in France. The problem is we need to know how old the bats actually were. So how do you study aging in bats? By the time a bat's about 3 months to 1 year, you cannot differentiate them between a baby, a 1-year-old, should I say, and a 30 or 40-year-old. Once the finger bones are fused, there's no way to age a bat. So what we do is we catch the entire colony of bats every single year. We put little microchips into the babies that indicate the year that they were caught and the year that they were born. They're released. The bats come back to the same route every single year to have their babies. And we, again, catch the whole colony. We develop these non-lethal sampling methods because we want these species to live long so we can study them, where we take less than 140 microliters of blood. We take a 3-millimeter biopsy punch from their wing membrane. We take microbiome swabs, we measure them, we weigh them, and we do this every single year. And we've done this now for the past 12 years. This will be our 12th year doing this. And what was interesting with this colony is they were studied by this conservation organization really for the past 20 years. And they've been all tagged since 2010. So we now know the age of these individuals. So we developed the methods to be able to ask the question, what happens to bats as they age? We also developed methods to be able to then take these 3-millimeter wing punches and reprogram them into bat iPSCs so that we can actually have a bat in a dish, because you cannot do experimental studies on wild bats. We need to be able to bring back the samples, make the tools required that we could test some of our hypotheses in the lab. And working with Thomas Waka and Marion Desauger, they came up with a new Yamayaka cocktail that was required to be able to reprogram bat cells. But we can do this now from 3mm wing punch. We're also developed the methods, again, to be able to grow fibroblasts from these wing punches. So we can actually do a whole bunch of cellular assays right across the entire population in the lab. So ex vivo analysis in the lab of these different bat cells. So we have the tools. But the interesting thing about bats, and this is something I want as a take-home message. This was a wonderful study done by my PhD student, Zhuang Zheng. But if you look here, this is the LQ, how long species can live for given their body size. Typically should be a correlation of 1. So ratio of maximum longevity to expected longevity given body size. And that's where humans fall. But look at bats. Bats can live so much longer, twice as long given their body size. So an LQ from a 4 to an LQ from 8 to 10. Humans, no matter what we do to humans, they do not have these naturally evolved longer health spans given body size, but the bats do. And what's quite interesting is that nature also with our bats has shorter lived bats and longer lived bats. So the experiments are already done where we can study shorter lived bats and the longest lived bats and see the changes that happen. But again, if you want to study extended health span and longevity, let's go look at organisms and species that have naturally evolved this, that are close to us, they're mammals, and these, of course, are our bats. So we developed the tools and techniques working with one population of these long-lived myotis bats in France. But as I showed you, there's a different aging ability across the order of bats. So we wanted to study different wild populations of bats and some captive colonies right across the spectrum of the LQ, from the longest lived to the shortest lived bats, and see what happens to them as they age. Now, what that means is an awful lot of field work. Tig, one of my PhD students over there, it's the fella in the blue shirt in this picture, went to travel all around the world, was in Panama, was in England, was in France, catching bats. A hard life, huh? And Tig also was involved really in driving the first question. So we wanted to look at how do the biomarkers of aging change with bats, from young, middle-aged, and older bats, and also individuals. And I'm going to give you the results, what we found with some of our telomere assays. And again, we used Cawthorne's RT-PCR method, comparing telomere length in a single copy gene versus a telomeric repeat. So telomeres, again, the TTA, GGG, that cap our chromosomes, maintain genomic stability. They shorten every time with cell reproduction because you can't add on to the 5' end. Once they're critically short, that cell stops dividing. It should be removed, but it can sit there becoming an old cell, emitting SASP, making our younger cells older. So again, telomeres are important. They're a major hallmark of aging. So what goes on in bats? So what we'd originally done this study with our myotis, the longest lived species of bats, longest lived genera of bats. And what we found that in the myotis genera, there was no relationship between telomere length and age. So telomeres did not shorten as would be expected given age in these myotis bats, these long-lived bats. We found that it didn't seem to be because the bats were expressing telomerase. So we take a little bit of wing, a little bit of blood, sequence the entire blood transcriptome, but found no evidence for the expression of telomerase. Now maybe this was because the RNA analysis isn't sensitive enough. There's not enough telomerase expressed or TERT expressed to be above the level of error. We also did a whole bunch of genomic and transcriptomic analysis, and we thought that DNA repair may be enhanced in these long-lived bats, and maybe this, and telomere maintenance, was allowing these bats to have their telomeres not shorten with age. So this initial study was done when the bats were 7+ years of age. Longitudinal studies, you have to keep doing them. So we kept doing this. Again, went to our 12th year. We added lots more individuals to this, and what you can see, as we add more and more older individuals, you find that telomere length still, there's no correlation with age in myotis. We have a whole bunch of individuals that were actually caught as adults. So we don't know their age, so they're 1+. And then we catch them year after year after year after year. We have to estimate their age. And even when you add lots more individuals, you still do not find any correlation with telomere length and age with myotis. What about Rhinolophus firmiquinum? The next species down, found in England. This is a wonderful long-term marker capture study of Roger Ransom in the UK. He's been doing this, I think, since he's 16. He's now in his 80s, very fit, very healthy, holds the Guinness World Record for the longest single marker capture study of an individual. And he's been catching and sampling these horseshoe bats, greater horseshoe bats, Rhinolophus firmaquinum, for that length of time. So there's over 2,000 individuals here. And these are sampled when they're born, sampled afterwards, continue going on. Again, we extract the DNA, estimate telomere, relative telomere length. And what you find is that telomeres, there is a correlation with telomere shortening with age in these greater horseshoe bats. We moved on to Rosetus egyptiatus, next bat down in our longevity spectrum. This is a zoocope bat. Again, you find there is a correlation with age, but it's a non-linear correlation with age. Phylostomus discolor, next species down on the LQ, you do find this linear shortening with age in these bats. And then molossus. This is the shortest lived bat on record. When we started the study, this bat, really, the mean age is about 1 or 2, and the maximum longevity of these species was thought to be 4 or 5. So we initially did this, and what you'll find, oh, look, there's 200 individuals. Potentially, there's a correlation shortening with age. But then Ty kept going back to Panama, kept catching these bats. We increased it. Age, maximum lifespan was now 6. Got lots more individuals who found, oh, if you add more, their telomere length, it's not shortening. But then this is the problem with field work and with nature. When you keep studying and keep catching them, we are able to now recapture individuals and double their life expectancy simply because we maintained the study. They can live much longer. But I want you to have a look at this. So we have still the majority of species. You'll find you'll catch 1 year, 2-year-old babies, but there's a dearth of individuals between 4 and 10. But telomeres, they're not shortening with age as would be expected. What about the effect of sex? Is there any difference in this? And what we found when you looked at all of our different populations, our different species, there was none of them showed any difference between male and females, apart from Rosetus. And these are these big fruit bats that were in the zoo. So there is a difference, male and female difference here. Let's remind ourselves, what's the effect of age? Well, in myotis are bats with the highest LQ, no significant of shortening with age. It's maintained, it's what we expected. Next one down in the LQ is Rhinolophus firmaquinum. You'll find there is a significant shortening with age. So in these species that are still quite long-lived, given their body size, telomeres don't seem to be as important. With Rosetus egyptiatus, there seems to be an increase and then a decrease with age. So there's a significant, a non-linear relationship with age. Phylostomus discolor is doing what we'd expect it to do. Telomeres are shortening with age. But then there's our molossus. And we were looking at this, and Tig said to me in our last lab meeting, maybe this shows the fact that if this species, so the majority of them are dying they're dying young, they're dying at 1 or 2, maybe in order to be a super centurion, you have to be able to maintain your telomere if you're molossus. So we're looking at that. But the question is, how are myotis? How are they maintaining their telomeres? What are the bats doing? So we wanted to then go and see what was going on. And so we couldn't find any telomerase expressions. Are they using alternative telomere lengthening? What are they doing? What's happening in the Rosetus for the first part of their life as they're increasing telomeres? So this is work done with Vera Gorbanova, whereby they were able to get the bat fibroblasts and do a trap assay, which measures the activity of telomerase. And doing this, which is different than doing these RNA transcriptome analysis, found that yes, indeed, in the myotis bats and partially in the Rosetus bats, there was evidence of telomerase activity. So these bats are able to extend and maintain their telomeres, potentially through the use of telomerase, so we're looking into this. But what's very interesting is that still they're doing this, but there's little to no evidence of cancer in any of these bats. So Vera and Andre and Fatima also did a whole bunch of assays and said, all right, how are they doing this? And they tried to make bat cells cancerous and malignant. And what you can do is you can knock in oncogenes, knock out DNA, tumor suppressors, and see how easy is it to make a bat cell cancerous. And the answer is it's extremely easy to make a bat cell cancerous. Indeed, you just knock out P53, and you can make them malignant, which is completely different than us in whales and very counter to what we expected. And I find this very, very interesting. This means that what bats have evolved, they've evolved ways to be able to somehow suppress tumors. Maybe it's not in the same way we're doing it. Could they have a form of immune surveillance? Do they have a much better way of being able to manage their cell regulation, their DNA repair, so they're not getting tumors? Or are they able to remove them before they become a problem? So we're looking at that. But then we also have our blood, and we wanted to ask, well, what happens in bats as they age? What happens to their overall transcriptome? We want to see what changes in bats as they age. When you do this, you look at young, middle-aged, and older bats. This is their whole blood transcriptome. We sequence all of it. It's work done by Zijia Huang. What you'll find is the major difference between bats and other mammals is as bats age, they increase their ability to maintain their DNA. But we want to ask the question, let's add more bats in. So that was just with meiosis. What then happens? And again, we have the meiosis bats. Then we have the Rhinolophus bat, second one down, the LQ. The next one in is going to be our shortest-lived bat, the molossus, human and mouse. And this is work done by Sebastian Riquier. And again, you can see we have 152 meiosis, 46 Rhinolophus, 30 molossus, and so forth, human and mice. Just to give you an idea of our numbers, they're still small at the moment. So what happened? So when you look at this, again, you're looking at these go pathways that change. And what you'll find is, again, your bats are the first three over, humans are the middle, mice at the end. We looked at 700 humans from GTEC, so the reality is we hadn't quite sifted through these. These are males and females. Individuals are very sick. So we'll remove humans for a moment to make it a bit simpler. But what you find here is that actually, when you compare bats with mice, you see a huge difference. Increase in DNA repair, cell cycle regulation. You find this kind of decrease in immune response and inflammation transcripts when you look at the blood transcriptome and you compare bats with mice. So something interesting is going on here. So we need to look at that. We also are able to put our bats into a dish, so have our fibroblasts, where we can actually go and stress them and see what the response are like. And when you do this, is work done by Orland Alcock, what you can find is, so cadmium is a DNA stressor. And you stress these cells and you see how easy is it to kill them. And you measure proxy of their function using this LD50. And what you find when you look at this is you see in red the meiosis bats, the longest-lived bats, it's quite hard to kill them. They need a lot of cadmium, which means they potentially could be repairing this. But next branch down is the molossus, which was once supposedly a shorter-lived bat. And then you have the Rosetta. So it's not quite so simple, but the take-home message is that DNA repair is important for nearly all bats. So we were interested in what actually regulates this. And so what you'll find is that actually we've been searching for what is driving this. So Bjorn talked about this before. We're now looking at the DREAM complex. Is there somehow bats have modulated its expression? We're looking at microRNAs and long-coding RNAs to try and find the regulators that underlie this unique signature. And that's very important if we want to be able to translate. So we're working on this also. But how do they maintain their telomeres? Looking at the overall blood transcriptome, looking for these 6 genes that are really deeply involved in the shell-to-rim process, you can see there's a difference in expression. And what we're looking for is expression of, is there something different happening in the longer-lived bats? Is something different that's going on in the shorter-lived bats? As you see here in TERT, you got to look at the actual x-axis. It's not really that much expressed, so we're not at all surprised we didn't see those results. But you can see a difference. And the meiosis bats maintain their telomeres with age. POT1 is very important. We also looked at this genomic selectivity. So we again sequenced genomes of long-lived and short-lived bats. Tanya Lam is leading this in the States. And what we were interested in is looking at pairs of long-lived versus short-lived close sister taxa. And what we wanted to do is we looked at all of the 16,000 genes that we could find orthologs for across all bats, 27 different mammals, carnivores, humans, and so forth. And we wanted to look for evidence of selection in these genes. And we wanted to look for evidence of selections in these hallmarks of aging. And the first thing to say that there's much higher level of selection in genes that are involved in the hallmarks of aging in bats versus other mammals. You can see here when you can look at pairs, just looking the pattern down these columns, these are short-lived versus longer-lived bats, there's different signatures. you find this selection happening on the ancestral branch leading to bats of genes involved in telomere maintenance. And that really, meiosis is showing some strange selection. So using comparative genomics can allow us to have a different lens and uncover different pathways that these bats have evolved to potentially allow them mitigate the damage of living. But also, just to finalize on this, we also were interested in looking at how are the bats living with these diseases? How are they dampening inflammation as they age? So as you can look at genomes, what you'll find is that there really are these very, very unique signatures in bats. They have lost their PIN genes that make an inflammasome. They have selection happening in a lot of the inflammatory genes. They've loss of these genes. They have selection happening on the non-inflammatory immune genes. So there's this unique modulation in the bat innate immune system. And indeed, we sequenced potential reservoirs of the SARS-CoV-2 and SARS-like coronaviruses in the Southeast Asian horseshoe bats, did a whole series of analyses I showed you before with the genomes. So again, looking for genes that are important, that change in individuals that have survived SARS-CoV-2. What are the networks and genes important for SARS-CoV-2? And what you find when you look here and you look at the ancestral bat branch, there's huge selection in these genes. And that bats are showing a change in these innate immune genes, both at the inflammatory and the anti-inflammatory pathways. So you can find important genes, and indeed, Matayan is going to be talking about this very, very cool paper where they use comparative genomics to find that the bat ASK2 gene is different to all the mammals, different to humans, and indeed, when you knock it into a mouse, that mouse can survive viral infections and also live healthier longer, but it's the next talk. So, not finished. So the question is why? Why have bats, what have they done? Why have they evolved this? So again, one of the hypotheses that flight is so very, very metabolically costly, that bats have had to evolve mechanisms to deal with the deleterious consequence of this high metabolic rate. They produce loads of ROS, breaks up DNA. They have to be able to fix and remove DNA damage, fix and remove protein and cellular damage. They have to be able to modulate their innate immune system so it is not getting overexcited by this constant sterile inflammation. This has led to unexpected longevity and a tolerance of pathogens. And we are very lucky because last year I pitched the proposal that we had for an ERC Synergy grant. We got it funded. And whereby, come and join us. It's a miracle. What we want to do is we want to bring together the study of wild bats, long-lived, shorter-lived wild bats and captive colonies. And we want to do a whole bunch of comparative genomics and redevelop the cellular assays and ask the question, what have bats evolved to allow them deal with the damage of living and deal with the damage of disease? And we want to integrate this all. So I need your brilliant minds to come together and think, how are we going to integrate this very heterogeneous data? To find the regulators that allow bats to have extended health span and also disease tolerance. And then we're going to knock them in to C. elegans. We're going to make bat worms and we're going to make bat mice and show that our predictors actually work. That's the plan. So we want to learn from bats, and I hope I've convinced you that we actually can. We want to go from the cave to the bench to the books and to the bedside. That's our plan. And just to leave you with this beauty. So she's still alive. This is our longest lived bat. She's now, I believe, 15 plus. She's still actually there. Just to show you that, so what have they evolved? They seem to evolve this very unique ability to repair their DNA, repair the code. They've evolved a way to be able to maintain this immune stasis. They're very good at going from a perturbed state back down to a nice homeostatic state. And studying bats will give us unique and new insight. And look at this. So this is all slowed down. But here she is flying. They've got to fly. They've got to echolocate. They have these unique health spans. And I want to say thank you so much to my brilliant lab that make it possible. Thank you to all our collaborators. And I apologize, it's a very sensitive clicker. Thank you.
Well, thank you so much, Emma. Questions for Emma? At the front here? Yep. Okay. Yeah, go on first.
Okay. So have you considered whether it would be interesting to compare all this data on bats to birds, which are also having to deal with, intense metabolisms?
Yeah, I absolutely have. I wrote a big grant to get it, and they laughed at me. So it didn't work. So we need to do that study. So what we've done, we've gone and we've looked at some of the comparative genomic analysis in the likes of the longer-lived parrots, in the longer-lived birds, and the albatross and so forth. Do you find the same signature? We haven't seen that yet. So we haven't seen something that would be a nice smoking gun that's independent phylogenetic contrast. But I think we need to really do this. Something's interesting, even, so with the birds, they also seem to modulate innate immune response. But flight is really, really important, and when there was this big macroecology study done, they had anything that flew was living way longer than expected, and that included the birds. So I think there's loads of scope to do this. So the answer is, I'd like to.
Thank
Just
you.
need money.
Yep.
Oh, that was just so great, Emma. It's always such a pleasure to watch you present. So in our work, IL-1 beta keeps coming up again and again as a target to deplete. It seems to be involved in all of the inflammatory diseases, many chronic, many acute, sepsis, cytokine release syndrome, on and on. It went by very quickly, but I saw it. And can you speak about what you're learning about IL-1 beta in the bat?
We're finding that it's suppressed. It's not at the higher level. We need to go and interrogate that an awful lot more. And the question is, how is the IL-1 beta suppressed? What's going on?
So maybe that has to do with their longevity and their resilience, is their ability to suppress it.
Yes, and I think Mate is going to be talking along these lines wherever he is now, maybe in a bit more detail.
Any more? All right, let's do it. Thanks so much, Emma.